Hello, everybody. Now I'm going to present a novel subunit targeted vaccine against bovine viral diarrhea virus, development and field results. My name is Demian Bellido and I come from Argentina. Reproduction is key in cattle. To deliver calf is the main goal of the cattle production in beef industry and dairy farm. Why? Because in beef you need calves, so you can feed, they will grow, and then they will produce meat. So it's key to have the calves. If you don't have the calves, you don't have nothing to produce meat. And in dairy farms, the milking process begins after the calving. So if you don't have calving, you don't have milk. So in both industries, to deliver calves is a main issue. So reproduction is key in cattle production. And BVDV is the most diagnosed infection agent affecting cattle reproduction here in Argentina. I don't know, I think all over the globe is similar, but here in Argentina, in different labs, we have the same results. And an important thing about this virus is that if we see the period of reproduction from the breeding season to the delivery, BVDV is the most diagnosed agent in the breeding season, causing infertility, causing early mortality, and in the 3rd trimester of gestation, causing abortion, from early abortion to late abortion. So BVDV affects and disturbs the gestation in every period. So it's very important to control. And to control this virus, the most used strategy is to vaccinate. And vaccines, all over the world, there are two kinds of vaccines, line-modified vaccines or inactivated vaccines. Now here, we are presenting a new one. It's a new kind of vaccine. It's a subunit and targeted vaccine, and it's called BVDV block. It's a subunit vaccine because instead of using the whole virus, we use just a receptor, the E2 receptor. And it's targeted because we add to this E2 protein, we add a targeting molecule. It's a single chain antibody called APCH that directs the antigen, the vaccine, to antigen presenting cells. And why that's important? Why we choose this target to direct the vaccine? Because antigen presenting cells that are a subset of white cells, the dendritic cells is the most important one, but there you have macrophages and lymphocytes too. The dendritic cells are the one that triggers the adaptive immune response. So it's very important to activate these cells, this subset of cells, to have a good adaptive immune response. So what has one APC in common? How my vaccine or the vaccine will find this? We need something that this APC has and the rest of the body doesn't. And this molecule is the major histocompatibility complex type 2, the MHC class 2. All the vaccine, all the cells in the body has the MHC class 1, and ours has the MHC class 2. So this is the target to the vaccine. Our vaccine is directed to the MHC class 2. Going to this molecule is a way to direct it to the APC, and to direct it to the APC is a way to trigger the adaptive immune response. So it's a way to increase, to make more potent the antigen, to increase the immunogenicity. The subunit targeted vaccine is the E2 protein fused to the APCH, forming the APCH2 molecule. There's a single protein with both molecules, just one protein. I have a video to show it, how it works.
BioInnovo presents BVDV block. BVDV block is a new concept in technology that is based on the recombinant protein APCH-E2. APCH is a single-chain antibody which recognizes the antigen-presenting cells of the immune system and the E2 protein, which is the most important protein of the bovine viral diarrhea virus, since it mediates the entry of the virus into the cell. From the genome of bovine viral diarrhea virus, the E2 protein gene is isolated and combined with the APCH gene using modern genetic engineering techniques. For the synthesis of this new protein, the baculovirus expression system is used, a novel system that allows the generation of large amounts of complex proteins, which then undergo the necessary modifications to be recognized by the immune system. This process results in the generation of a large amount of high immunogenic APCH-E2 protein, which is the basis of the new targeted vaccine BVDV block. Thanks to its exclusive targeting strategy, the BVDV block's molecules go in search of the antigen-presenting cells of the immune system. These cells recognize, process, and present foreign antigens on their surface. Then they interact with lymphocytes to activate them and in this way start the adaptive immune response. The active lymphocytes generate a large amount of specific antibodies that are able to bind to the virus and thus neutralize the infection. BVDV block, the revolutionary tool for the fight against bovine viral diarrhea virus. BioInnovo, creating tomorrow's health today.
Well, that was a very nice explanation. So, as we saw, our vaccine works in a different way to all the other ones. Because in this case, the antigen goes in search of the immune system. In every vaccine, the immune system is the one that goes to recognize the antigen. So it works in a different way. It's like the mice chasing the cat. So our vaccine is directed go in search of the immune system. But it's better because the immune system is active. So the immune system goes in search of the vaccine and the vaccine goes in search of the immune system. So they work like a magnet. We have a very potent immune response because we have very good stimulation of the APC cells. So APCH-E2 is more effective vaccine, and to test that, we did in guinea pigs, we did a comparison between the E2 protein using several amounts of E2, and from 3 to 100 micrograms, and just 1.5 micrograms of antigen in the BVDV block composition. And what we saw in the guinea pigs is that those are neutralizing antibody that with just 1.5 micrograms, we have a better, more potent immune response than using 100 nanograms. So it's a very important immune response, and it's 66 times more efficient vaccine because 1.5 gram dose has the same response, even a little better than 100 nanograms. So it's 66 times more. It's a lot. But this was in guinea pig, and the question is, in cattle, does it work? Now we're going to present results. I'm going to present 2 results. And this is in beef cattle. We have also results in dairy farm, but for matter of time, I cannot present it here, but I hope next year I presented some dairy farm results. The first thing I'm going to present is the potency test. It was a registration trial performed in a governmental facility with 24 seronegative heifers, 8 received targeted vaccine, 8 the reference vaccine, and 8 placebo. Just 2 doses, day 0 and 30. Samples were collected at different times and were analyzed by seroneutralization assay. And the targeted vaccine induces a potent and neutralizing antibody response. As we can see in the graph, the vaccine at day 30 days post booster here in day 30, day 60, reached a neutralizing titer of 16,000, over 16,000, while the reference vaccine of Senasa, which has a traditional inactivated vaccine, a commercial traditional inactivated vaccine, reached 100. So here is 100, again it's 16,000. It's huge, huge difference. This is a threshold of protection. If you have antibodies above this threshold, you can say you are protected. The health will be more protected. So our vaccine is over this limit the whole year with just 2 doses, day 7 and 30. And you can say with the other vaccine it's going to go down here about like 120. And then another important thing of this trial is that in gray here are the negative animals, and they never serocompared. So here we have known virus circulation. So all the immune response that we saw is triggered by the vaccine. And now I'm going to present results from beef, but from commercial facilities. Well, 3 beef cattle commercial facilities in different states of Argentina, 100 animals per farm, 300 total, 50% received targeted vaccine and 50% in conventional vaccine. It's a commercial vaccine that has inactivated BVDV in the formulation. Here there is no placebo group. So I will show you the graph and you will see targeted and controlled. And you have to know that the control are vaccinated with a conventional vaccine. Samples were taken and the results were analyzed by ELISA. And what we found is that at day 0, we have like mixed titers. Some animals with high antibody titers and with negative or low, some with mild antibody titers. So there was a mixture. What we saw is that targeted vaccine induced long-lasting and protective immunity. If we compare the results of the targeted vaccine with the control group that received a conventional vaccine, we see that there is no big increasement in antibody levels in the control group, and there is a very important increasement in the targeted vaccine group, and it's maintained for the whole year. But that's the same. We separated in the animals who had high titer at the beginning and low titer at the beginning of the trial, so we can compare it in a better way. What we saw here is that when we analyze animal high antibody level at the beginning, is that the animals that receive the targeted vaccine increase the antibody level and then maintain into the end of the period. The control group has a little increase here and go down to the end of the year. And the most important part are the susceptible animals, and we saw that the targeted vaccine can protect them. To protect these animals is the key of the vaccination campaign. That is what we want. The other animals, the ones that start with high antibody levels, well, they will be protected. But what we want to protect are the low antibody titers animals. And what we saw is that animals that receive 2 doses of BVDV have a very, very nice cure here, a very potent immune response, as it maintains antibody level over the threshold of protection the whole year. Meanwhile, animals that receive the commercial vaccine, the conventional vaccine, has a little increase here and never reached the threshold of protection. So we can protect the animal that we need to protect. Conclusions. The targeted antigen APC-E2 is 66 times more immunogenic than the E2 protein alone. The targeted vaccine was able to elicit a potent and long-lasting immune response against BVDV, and it was much higher and last longer than the traditional vaccine in the clinical trial in the Potency Test. Negative and low titer animals at day 0 which is similar antibody levels like high antibody titers. And the most important that we can protect it, they was over the threshold of protection from the whole layer. And that's the objective of the vaccination. And the last conclusion is that this subunit targeted vaccine represents a novel, safe, and efficacious vaccine against BVDV. This is our stuff, and thank you, and I'll be happy to answer some questions.